Hello everybody, Namaskar. Now we are in the 10th module of the course, and the 5 lectures of this 10th module will be devoted on various aspects of plant based fermented foods and beverages. In today's lecture, that is lecture 26, we will discuss the concept and process basics and fundamentals of fermentation technology. Okay. The various aspects of fermentation technology that will be covered in today's lecture is, that is, what is fermentation, why it is needed, what are the different microorganisms and starter cultures used in the fermentation process, fermenters and various types of fermenters. Then we will also discuss something about heat and mass transfer during fermentation process, and finally fermentation process control and monitoring. Okay. Let us see what is the fermentation. The term fermentation derives from the Latin word meaning fervor. It means that is to ferment. Louis Pasteur, who was a French chemist, he discovered that yeasts convert sugar to alcohol and carbon dioxide during fermentation process. Campbell Platt in 1987 defined fermented foods as those foods which have been subjected to the action of microorganisms or enzymes, so that the desirable biochemical changes cause significant modification to the food. It also describes a form of energy yielding microbial metabolism, that is, fermentation is a form of energy yielding microbial metabolism in which an organic substrate, usually a carbohydrate, is incompletely oxidized and an organic carbohydrate acts as the electron acceptor. So, the fermentation is natural. It occurs whether you want it or you do not want it. However, if it is controlled, it is a very valuable process. It adds value to the product, but if it is uncontrolled, it will spoil the product. The food will be spoiled by fermentation. Okay. So, the fermentation process is environment friendly, it consumes less energy, produces less waste, and it is easy to manage under household conditions of low income communities as well as in the industrial scale for urbanized welfare societies. Fermentation is needed to enrich the human dietary through development of a wide diversity of flavors, aroma and texture in foods. Fermentation is a preservation technology. It preserves the food through lactic acid, alcoholic, acetic acid, alkaline fermentation processes, and even high salt fermentations are also in earlier classes we discussed about this. Also, there is enrichment of the food substrate biologically with the vitamins, protein, essential amino acids, salt, etc. These are all many such compounds are produced, are generated, synthesized by the microbial action inside the food. Fermentation also results some sort of detoxification of the food. There is a removal of anti-nutritional factors, etc. in the fermentation process. There is a decrease in the cooking times and fuel requirements in the fermented foods. Okay. The popular plant-based fermented foods and beverages are, can say that, vegetable-based like kimchi, mixed pickle, sauerkraut, gundruk, fruit-based product maybe wine, vinegar, cider, perry, brandy, etc. Chocolate, nata de cocoa, raki, these are cocoa-based fermented products. Even mead and methigin, they may be honey-based, or even tea-based fermented products include Perich tea, kombucha, jun tea, labet, goshicha, and so on. So, in the fermentation process, as discussed earlier in that definition, that is glucose is converted actually into pyruvate by either then this pyruvate it may go that is fermented oxidized further by the aspergillus into lactic acid, as in the happens in the soya sauce, or lactobacillus group of bacteria, it converts pyruvate into lactic acid. This is found in the cheese or yogurt, and it may be yeast fermentation like Saccharomyces cerevisiae, where it may result in the beer, ethanol and carbon dioxide, in the wine, fruit wines, etc. This pyruvate is converted into ethyl alcohol, or in some other product like bakery products, etc. Same yeast enzyme, it converts it into carbon dioxide is the major product. So, the fermentation types, mainly two types of fermentation are there, either submerged fermentation or solid state fermentation. Okay. So, in the submerged fermentation, organism grow beneath the surface of the medium, whereas in the solid state fermentation, organism grow on the surface of the medium. Okay. In the solid submerged, it is a liquid medium is generally used, whereas in the solid state fermentation, a solid medium, solid substrate is used. Okay. The culture medium is always free flowing in submerged fermentation, whereas in the solid state fermentation, culture medium does not flow freely. Water content of the medium is about 95% or above in the submerged fermentation, about 40 to 80% in the solid state fermentation. Nutrient distribution is even in the submerged fermentation, but in the solid state fermentation it is not even, maybe uneven. Inoculum ratio is generally low in the submerged fermentation and high in the solid state fermentation. So, the fermentation oxygen consumption depending upon the oxygen availability and its utilization, it may be anaerobic or aerobic fermentation. Aerobic fermentation is carried out under aerobic environment, that is in the presence of oxygen, and here glucose is completely oxidized, giving carbon dioxide and water, and a huge amount of maybe around 36 ATP molecules of ATP are generated from one glucose molecule. In the anaerobic fermentation, obviously there is a strict anaerobic conditions. Here even facultative anaerobes also sometimes they may manage to perform anaerobic fermentation. Okay. And there is a bacterial, and most of the bacterial are yeast fermentation which occur if absence of oxygen. This anaerobic fermentation may be lactic acid fermentation, where lactic dehydrogenase enzymes converts glucose into lactic acid, and then 2 moles of ATP is generated. In the case of alcoholic fermentation, that is alcohol dehydrogenase enzyme, yeast, it converts glucose into this ethyl alcohol plus carbon dioxide, and also some energy in the form of ATP is released. As far as the starter culture concept is concerned, this starter culture concept developed to have a better control over the fermentation process to produce desired quality or good quality product. Starter culture is defined as the strains of microorganisms which are selected with stable features to produce desirable characters in the product under controlled conditions. They may be that the microorganisms like bacteria, yeast and fungi are the starter culture, and then proper temperature, that is all these microorganisms they have some optimum temperature requirement for their growth. So, that proper temperature should be maintained, that is like mesophilic or thermophilic, and then a number of the strain is also important, whether it is the single bacteria or multi bacteria and the mixed mode and so on. Okay. Then let us discuss about the fermenter. It is the heart of the fermentation process. Okay. A fermenter, it is basically a bioreactor where all the control operations etc. are provided with instrumentation and control. The ports etc. which are provided, and it facilitates the process like this. It gives a contamination free environment. You will be able to control the specific temperature maintenance and control, maintenance of agitation and aeration, pH control, monitoring of the dissolved oxygen, ports for nutrient and reagent feeding, ports for inoculation and sampling, fittings, etc., geometry for scale up, and minimize liquid loss and growth facility for the wide range of organisms. So, all these elements are provided in the fermenter or bioreactor. You can see here in the figure. Okay. Then the basic requirements of a fermenter, that is the fermenter, that is it's a material that is used for the construction, should withstand the steam sterilization, it should be non-toxic, non-corrosive, must prelude foreign contamination, etc. If there are any, that is to be able to be sterilized. Optimal mixing with low uniform shear achieved by proper designing of the agitator and aerator. Adequate mass transfer, that is oxygen, achieved by monitoring the speed of the agitator and gentle heat transfer. Anti-foam agents like anti-animal vegetable oil, lard oil, corn oil, soybean oil, etc. are used in controlling the foam, because many sometimes when you agitate, some foam is generated. So, anti-foaming agents may be added. So, there should be arrangement for that. There should be port for pH, temperature, dissolve O2 or sensors for regulation, etc. Feeding for substrate with prevention of under or overdosing by proper feed ports and monitoring suspensions of solids, etc. These are the provided. So, these are the basic requirement of any fermenter. This all the fermenters should have these arrangements. Okay. So, normally the components of the fermenters and their functions are which are there, they are given, this is given in this table. That is the major components. It is a vessel, it may be glass jacketed steel vessel with ports, various for output, input, etc. probes. Then drive motor, drive shaft, impeller, aerator, baffles which prevent sedimentation on sides and it ensures proper mixing. Then feed pumps which regulates the flow rate of additives, peristatic pump for our fixed speed pump which is used for continuous sampling, three-way inlet to insert different probes or valves that regulates and controls the flow of the liquid and gases, etc. So, all these are the components of the fermenter. Then the various ports which are used in the fermenters, you can see here in this figure, like pH probe for measuring and monitoring the pH of the medium, temperature probe, foam probe, dissolved oxygen probe, or even level probe, that is the level which could indicate the measure the level of the medium inside the reaction vessel. Then sampling port to obtain samples during the process at proper intervals, so as to check the fermentation process and level of alcohol or acetic acid that is produced, which facilitates that stoppage of the fermentation process. Then rotameters which measures the flow rate of the air and the liquid, pressure gauge, it measures pressure inside the fermenter. So, all these ports, etc. are provided in the modern fermenters. Okay. Stages in the fermentation process, that is there are 3 stages, stage 1, stage 2 and stage 3. Stage 1 is basically upstream processing, which includes preparation of the fermentation medium, sterilization of air and the fermentation medium, and finally inoculation of the fermenter. So, these are the upstream processing. Then the fermentation, actual fermentation process, which involves the conversion of the substrate to desired product with the help of biological agents like microorganisms, etc. Okay. So, that is in the bioreactor when the reaction normally takes place, that is actual fermentation process. And then finally, the downstream processing, it involves separation of cells from the fermentation broth, purification and concentration of the desired product, and the waste disposal or recycle. Okay. The fermentation media is the main component, very important. Okay. Most fermentation requires that the liquid media, okay, and it is often known as broth. The fermentation media should be rich in the nutrients required for the growth of the microorganism, and it should fulfill the technical objectives of the process. The media should contain a carbon source, a nitrogen source, energy source, and the desired micronutrient required for the utilization and growth of the microorganism, and then finally for the production of the products. So, the characteristics of a good media include it should be cheap, should be easily available, okay, it should maximize the productivity, should be required in least quantity, and should minimize the formation of undesirable products. Generally, the carbon sources in the fermentation media includes molasses, malt extract, starch or dextrin or cellulose, whey, methanol or ethanol, hydromolasses or sulfate waste of the liquor, etc. So, either bio waste or from various organic sources, all these, they like for example, molasses is a byproduct of the sugar industry. So, it is a cheap source of the carbohydrate. It contains also nitrogenous substances, vitamins, trace elements, etc. Sulfide waste, it is a spent sulfide liquor from the paper pulp industry. It is used in the production of ethanol by Saccharomyces cerevisiae, and the growth of Tarula stilis is as a feed. So, these are the various carbon sources. The various nitrogen sources may be inorganic form of nitrogen like ammonium, salts or ammonia, etc. Then urea, it is an organic source of nitrogen. Then corn strip liquor, yeast extract, soya meal, peptones, etc. These are the various. So, a suitable source of the nitrogen, carbon or nutrients, etc. energy source, this should be provided in the microbial media. Okay. Then we will discuss the type of the fermenters or bioreactors. They are of different types like continuous stirred tank bioreactor, airlift bioreactor, packed bed bioreactor, fluidized bed bioreactor, photobioreactor or a membrane bioreactor. The continuous stirred tank bioreactor, as you can see here in the figure, it consists of a vessel, pipes, agitator shafts, impellers and motors. It is most commonly used fermenter. The main target of the desired product enlists the cell or primary metabolites which mostly acquire the microorganism like yeast or bacteria. The stirred tank reactors offer excellent mixing and reasonably good mass transfer rate. You see here, these are the arrangement provided for stirring the content inside the tank. The cost of operation is lower and the reactors can be used with a variety of microbial species. Then airlift bioreactor, you can see here the medium of the vessel consists of draft tube through which the air is pumped inside. See here, if the air is pumped, this might create a bubble in the medium which ultimately helps in rising up through the baffle tube and drags the surrounding fluid as well. This in the process stirs up the content by air. There are two types of airlift bioreactors. One is the internal loop type and the external loop type. Internal loop airlift bioreactor has a single container with a draft carrying out the fermentation process, whereas the external loop airlift bioreactor has an external loop, as the name itself indicates, to separate samples or liquid in different channels carrying out the fermentation process. The packed bed bioreactors, they are tubular reactors having a packed bed with biocatalyst, okay, that is immobilized enzyme or microbial cell. You see here packed bed with the biocatalyst in the reactor. Okay. So, a nutrient broth continuously flows over the immobilized biocatalyst. After that, a product releases into the fluid at the bottom of the container. You can see here in the figure. Okay. And finally, it can be removed. Here, the flow of the liquid can be either upward or downward, as the case may be. Fluidized bed bioreactors, okay, it is a combination of stirred tank and packed bed system having higher volumetric capacity. The top port is the more expanded to reduce the velocity of the fluid, whereas the bottom port is slightly narrow. You can see here in the figure. It is designed in such a way where the solid retain inside the vessel and liquid flows out, generally operated in co-current upflow with liquid as continuous phase and other more unusual configuration like the inverse 3-phase fluidized bed or gas solid fluidized bed. These are the much more importance. Okay. The smaller particle size facilitate higher mass transfer and the better mixing in the see here that it is materials they are kept in the pharma bubble or fluidized, okay, and it gives intimate mixing. So, the reaction is better. Then photobioreactors, this reactor works on light energy either through the sunlight or artificial light and have the temperature of around 25 to 40 degrees Celsius in the reaction medium. They are mainly used for algae production, for keratin production, astaxanthin production, etc. They are made of transparent glass or plastic tubes. These tubes act as solar trappers and culture transfer inside through a centrifugal pump. It is cooled and temperature and when the temperature reaches about 40 degrees Celsius. The microorganism grow in the presence of the solar light and then product forms during the night. Membrane bioreactor is here the suspended biomass and solid removal is done by ultra and microfiltration membranes. These membranes mostly uses the polysulfonate, polyamide or cellulose acetate membrane. They are used for alcoholic fermentation, solvent organic acid production or even that is wastewater treatment, etc. The advantages of the membrane bioreactors include that there is a loss of the energy, loss of the enzyme is minimum, effluent quality is high and the effluent is properly disinfected from all the pathogenic microorganisms. However, these are costly, it consumes more energy, it has limited aeration capacity and the fouling of the membrane is a major disadvantage. Now, let us see some heat and mass transfer phenomena during the fermentation. First, mass transfer. Here the diffusion plays major role in the bioprocess and the fixed law of diffusion, that is J is equal to minus dAB dCA by dy, where J is the molar flux component of A in y direction, dAb is the molecular diffusivity of molecules, Ca is the concentration of A and y is the distance of the diffusion. So, this fixed law of diffusion holds good here. Okay. So, it is that is scale of mixing, solid phase reaction and mass transfer across a boundary membrane. Then the oxygen transfer to the gas bubble to the fermentation broth, penicillin recovery from the aqueous to the organic liquid or glucose transfer from liquid medium to the mold pellets are typical examples of the mass transfer across a phase boundary. Okay. The general mass balance equation like dVc by dt is equal to V Q dash plus Q, where Q is the volumetric transfer rate, Q dash is the volumetric transfer rate and Q is the volumetric production rate. So, this V multiplied by volumetric transfer rate plus volumetric production rate plus BFCF minus BECE, considering that in the reaction medium that is the culture is flowing, that is at a rate BF, that is flow inflow BF and concentration of the flowing substrate is the C. It is outflow is the VE and C, and V and C are the volume and the concentration of liquid inside the reaction medium. So, this general mass balance equation is hold good and this can be calculated. Then heat transfer, that is heat is generated in submerged microbial system by the metabolic activities of the microorganism and the mechanical work performed by the agitator and by the gas bubbles as they expand. So, for geometrically similar vessels the volume increases as the third power of the linear dimension, whereas the surface area increases only as the second power. Transfer of heat between the microorganism and the surface of the vessel is somewhat analogous to the mass transfer, and direct determination of cell to fluid heat transfer coefficient is much more difficult than the determination of mass transfer coefficient. The fermentation process, that is the mode of the fermentation, is dependent on the relation of the consumption of substrate to the biomass and the product. It may be batch fermentation, it may be continuous fermentation, or it may be a fed batch fermentation. In a batch fermentation, that is, it involves a sequence of operations from development of inoculum from a stock culture to seed or a production of fermenter. So, a number of stages may be involved, but the production stage is usually performed in a single fermenter. Multi-stage system has the advantage of increased productivity. That is, required fermentation time varies with the hours to week depending on the conversion attempted and the conditions that are used. Growth rate is normally uncontrolled and it is highest at the start of the fermentation. So, the productivity in the batch fermentation process, that is the final concentration of the product or biomass, that is form P, that is divided by the time of batch fermentation plus turnaround time. So, this gives the productivity of batch fermentation. The turnaround time is the time for emptying, cleaning and sterilizing and finally refilling the reactor. The continuous fermentation is open system to maintain cells in a state of balanced growth by continuously adding the fresh medium and removing the culture medium at the same time. You can see in the both there is a continuous system where continuously there is the feed is added and a particular concentration and maintained, and then continuously the product is good. So, the rate of the product goes out at the rate of the feed added, it's some balance is maintained. Okay. And there is other system where the with cell recycle system, it is also that in the continuous, but in this when the product goes out, there is some underdeveloped cells etc. They are separated and they are recirculated. So, this is a cell recycle system. So, there are two modes of operation like chemostats, where most widely used apparatus for studying microorganism under constant environment conditions. It uses low dilution rate, and oxostates are the normally either turbitostates which controls the feed rate, pH state, oxostates, the feed rate is regulated by measurement of control of the pH of the fermentation medium, or nutristat, it involves regulation of the feed rate to maintain the residual substrate concentration during the fermentation process. Okay. Then productivity of a continuous fermentation process, that is the calculated by multiplying the dilution rate D by the concentration of the product in the outlet stream. That is the cell productivity is dx in terms of kg cells per cubic meter per hour, or product productivity is the D multiplied by P, that is the kg of the product per cubic meter per hour. So, the productivity in the continuous fermentation process, it can be either in the terms of cell or product, whatever you say, and there is a no turnaround time, because in the continuous fermentation emptying, cleaning, sterilizing, refilling etc. are not a problem, not done. Then in the fed batch fermentation, it is a technique in between the batch and continuous fermentation. It is suitable for non-growth associated products. Fermentation involves two phases, growth phase and production phase. So, fed batch fermentation is well suited for producing the product or cells when the substrate is inhibitory and there is a need to maintain low substrate concentration to avoid the cell being inhibited, or when the product or biomass yield at low substrate concentrations are high, as in the case of Baker's yeast, antibiotic production etc. The productivity in the fed batch fermentation process, it is better than compared to the batch mode of operation. In batch fermentation of Saccharomyces cerevisiae, a dry cell weight of 1 gram per liter was reported, whereas in the fed batch mode, the respiratory quotient RQ as the control parameter for glucose feed, a dry cell weight of around 31 to 56 gram per liter was obtained. So, the productivity in the fed batch fermentation medium is better than that of the batch medium. So, the process monitoring and control is very important in the fermentation process, because if this is not maintained, for example, if the temperature is not maintained, if the in case of aerobic fermentation, oxygen level, and particularly in the larger reactors throughout in the liquid, that is oxygen, in the anaerobic fermentation there should not be completely at all, even the pH and all those. So, most of the modern fermenters, they are provided with all, that is there is a shaft power for the torsion dynamitron, strain in gas measurement, there are filter assembly provided, air compressor, there is exit gas cylinders like oxygen paramagnetic, carbon dioxide regulator, mass spectrophotometer or pressure gauge, agitator speed, foam detection meter, etc. Okay. That gas electrode, pH, temperature, thermocouple control. So, and even the most modern fermenters, they are properly controlled, and even that is a computer control that are there. The computer data and station are provided with the computer system. So, mostly the physical variable like temperature, impeller speed, aeration rate, pressure, chemical variable like pH, dissolved oxygen, dissolved carbon dioxide, redox potential, and biomass, oxygen uptake rate, carbon dioxide production rate, respiration quotient, all these things are properly monitored and controlled, and the modern aerators should be provided with the necessary arrangement to maintain these parameters, etc., in order to get the product desired, or to control the process in such a way that the desirable microorganisms are only growing, that and the desired product is produced with the desired characteristics, and undesirable or wild growth of the microorganisms, etc., is controlled. So, that is very important. Otherwise, if it is the fermentation process is not controlled properly, then wild microorganism, spoil microorganism or wild yeast, etc., they may grow and they may spoil the product. So, I will summarize this lecture, that is by saying that yes, there are 2-3 important aspects in the fermentation process, that is there should be a proper well defined substrate and a proper bacterial or microbial or yeast or mold proper culture, and this culture required for, and the selection of the culture and the substrate, like we take the juice, maybe grape juice, and then Saccharomyces cerevisiae yeast in proper form, and then before adding the yeast, this grape juice is calculated, its sugar content, pH, etc. So, proper condition of the proper characteristics of this, that is you should ensure that in the substrate proper quantity of ingredients are there, and then you provide the desired conditions for the bacteria or yeast or other microorganism to grow, and at optimum requirement of pH, temperature, other things is maintained, so that you get a proper level of the product with desired characteristics. So, that is fermentation media should be rich in all the nutrients and carbon and nitrogen source, and it should, the fermenter should be properly designed and controlled to give the desired product. So, this is all, these are the references of used in this lecture. Finally, thank you very much for your patience. Thank you.